this is Liz, and I'm going to talk to you about how we can assess ecosystems, see what's there and how it's distributed. Population sizes are interesting to ecologists, and there are various experimental tools that we can use to look at ecosystems and the populations within them. One of these is Quadrats. I don't know if you've used these in school. They're the square frames that can be used to estimate population sizes. So you might have checked them about on the grass and counted how many flowers or plants were inside it. Another one is Transects. And transects can be used to investigate the effect of a factor on the distribution of a species. A transect line is a piece of tape that is stretched across the habitat of interest, and quadrats are placed at regular intervals along the transect line. So if you've got a big area that you want to cover, you can use a transect to see if the species that are present change throughout that habitat. So quadrats are the square frames used for estimating population sizes, and transect lines are the lengths of tape stretched across a habitat to investigate the effect of a factor on a species' distribution. When discussing organism abundance, we can use lots of different terms. So abundance is the number of species that there are. So we might look at the median, which is the middle value in a numerically ordered series. So you put all the numbers in order, and the median is the middle number. Another thing we might look at is the mean. The arithmetic mean is the average value of a series of numbers. The mean is calculated by adding up all of the values and then dividing by the number of recordings that have been made. We also might look at the mode, which is the most common value in a series, or the most fashionable one, if you want to remember it like that. So those are the mean, the median, and the mode. So how can we use quadrats to estimate a population size? I'll just run you through the steps now. So first, we divide the habitat up into a series of quadrat-sized cells. Then randomly select a given number of cells and go out into the habitat and place the quadrat in these positions. To evaluate the quadrat contents, either count the number of individual organisms of interest or record the percentage of the quadrat taken up by an organism. The samples from the quadrat are then used to estimate the total population in a given area. And how do we calculate the population? So there's an equation you can use where population equals the number counted times by the total survey area divided by the area sampled. That's the equation you need to know. And that sums up how you can assess ecosystems. So you can use quadrats, and you can use transects, and then you can calculate all sorts of statistics such as the mean, the median, and the mode.